Hi, and welcome to my OCRA A-Level Biology revision session with me, Christine. So today's lesson I want to look at Module 6 and specifically gene mutation. So before we get started, let's just take a step back and remind ourselves about the genetic code. So the genetic code is our DNA sequence, which is going to code for this sequence of amino acids. So we have to remind ourselves that we have what's known as a triplet code. So we have 3 DNA bases that code for an amino acid. For example, the codon TAC is going to code for methionine, the start codon, therefore the first amino acid in our chain. We also need to remember that the genetic code is non-overlapping. Non-overlapping, so it's always read in the 3 triplet bases, and it is universal, so it's found in all organisms because they all use exactly the same code. The other part we need to remind ourselves is about our degenerate code, that we can have more than one triplet code that codes for a very specific amino acid. So as we can see here, guanine, guanine, adenine codes for proline, but so does guanine, guanine, thymine. And we need to remind ourselves of transcription that occurs within the nucleus. So where we have RNA polymerase that's going to form those phosphodiester bonds, creating our pre-mRNA, our single strand, which will then be spliced to provide us with our mature RNA. And that can leave through the nuclear pores into the cytoplasm, where it will then bind to the ribosomes for translation to occur, where our tRNA will bring along the corresponding amino acid because of the codon being complementary with the anticodon on the tRNA molecule. So if you haven't already done so, do check out my videos on transcription and translation. But what we have to remind ourselves is the primary structure that was built with that translation occurring led to our sequence of amino acids. So the code, the genetic code, is the DNA sequence, which will therefore lead to this sequence of amino acids, and we call that our primary structure. Now, our amino acids are obviously held together by those peptide bonds. Our secondary structure, we've got these interactions, either alpha helix or the beta pleated sheets. And then we have our tertiary structure, how they fold and bend because of those interactions of those R groups. So that sequence is going to determine the secondary structure. It's then going to determine the tertiary structure. And we obviously have our quaternary structure where they are coming together, these different protein subunits, 2 or more, giving us our quaternary structure. So how does gene mutation affect protein structure? So what we have to understand is that what causes a mutation is spontaneous. It can actually happen during DNA replication in our S phase. So let's just remind ourselves that that is a semi-conservative replication process. We have our DNA strand that is going to be unzipped. We have our new bases which are going to form complementary base pairs, and we're going to end up with one molecule which has one parent strand and one new strand, hence its name, semi-conservative replication. Well, spontaneous mutations occur during this replication process, and what does that mean? Well, a mutation is basically an unpredictable change in the DNA base sequence of an organism. If we're talking about one nucleotide, we say this is a point mutation. Now, there are different types of gene mutation, so let's start with a base substitution. So here you can see I have my DNA base sequence. I have a TCACACGAGTCA. Remember, in the exam you should be using the full names unless instructed otherwise. Well, let's just imagine that we have a base substitution. So instead of the sequence that we originally had, instead of the guanine, we've now got a thymine in that position. Well, that is going to have a few consequences potentially. So what are the consequences? Well, it might be what's known as a silent consequence. Basically, it's going to code for the same amino acid and therefore have no effect. Well, that is the degenerate nature. So we know that the degenerate code, we have the same codons could code for exactly the same amino acid because there are only 20 amino acid variants and there are 64 possible combinations. Well, 3 of those are stop codons, so therefore 61 of them could be multiple for that same amino acid. So that could result in a silent consequence. We've had a base substitution, but it could be silent because we've got exactly the same amino acid in our primary sequence. Or it could be that this mutation actually occurs in an intron, which is known as a non-coding region. It could be what's known as a missense consequence. So basically, instead of it being the same amino acid this time, we've now got a different amino acid. And that different amino acid could be beneficial. It could help in whatever the role that protein has to play. It could be harmful in the fact that it could cause that protein to be non-functional, or it could be silent. The amino acid that's different is actually away from the functional part of the protein. So when I say that, if you think about an enzyme, for example, it could be that this different amino acid is actually nowhere near the active site, which is the functional part of that protein. So yes, we have a different amino acid, but it might not actually affect the role in which the protein has to play. They love throwing that one in there sometimes. And the last one of a consequence is a nonsense. So a nonsense is basically this introduction of a stop codon. If we're introducing a stop codon, that could have a negative or harmful effect on the protein's function. So let's just give an example of this one then. So if we look at hemoglobin, normal hemoglobin and the sickle cell hemoglobin, which is known as a mutant hemoglobin. Well, what you need to know is there is one base substitution. As you can see here, the GAG, so guanine, adenine, guanine codon on the 6th amino acid sequence. So I've missed out a couple before, but this is at the 6th amino acid position. Guanine, adenine, guanine was the original codon. In the mutant hemoglobin for sickle cell, we now have guanine, thymine, guanine. Well, because we have got this point substitution within the gene, what's happened is instead of us having the hydrophilic amino acid glutamic acid, we now have a hydrophobic amino acid valine. So we know that hemoglobin is this globular protein made up of 4 subunits. Therefore, we know its quaternary structure, 2 alpha chains, 2 beta chains. We know that the sickle cell anemia is actually caused by this base substitution mutation on the beta chain, which results in that different shaped protein due to the valine on the 6th position. The hemoglobin is now less soluble. So when the partial pressure of oxygen is low at respiring tissue, that hydrophobic valine actually binds to adjacent hemoglobin molecules. And that causes the deforming of the red blood cells to give a sickle shape. So this is an example of the point substitution, therefore having a missense consequence and therefore having a harmful effect. So let's look at another type of mutation and gene mutation, which is a base insertion. So if my base inserts, as we've just shown here, my thymine got inserted, what we resulted was a frame shift. So all of the other DNA base sequence were shifted down the frame. Now remember, we have our triplet code, we have non-overlapping. Therefore, this frame shift is disrupting the genetic code. That's leading to the production of a completely different protein or a non-functional one, which may cause various effects in an organism. So we don't really know what those effects might be, but when there is an insertion, that frame shift is going to disrupt the genetic code. And that'd be the same if we deleted a base out. So if we take the guanine out here, this time again, we've got another frame shift, but it's going in the opposite direction. So again, we are disrupting the genetic code, leading to this production of a different protein, a non-functional one. So the rates of mutation are obviously spontaneous with DNA replication, but we can increase them with mutagens. So physical mutagens like X-rays, ionizing, radiation can actually result in the breaking of one or both of the strands. Now, what tends to happen is as those strands are being repaired, those mutations will occur during that reparation. So that could be one way in which we increase the rate of those mutations. Another one is a chemical mutagen. So if we are talking about carcinogens are chemical mutagens. Now, one of those could be what's known as a deaminating agent. Well, what does deamination mean? Deamination means the removal of the amine group. Well, if we remove the amine group from cytosine, what we result in is having a different base. We've now got uracil. So we've still got a pyrimidine base, but this time, if you think about that complementary base pairing, that is going to be affected. Before, the cytosine would have paired with guanine, but now because of this deaminating agent, instead of it being cytosine with guanine, we've now got uracil with adenine. Well, therefore, we are now changing that base sequence. So that could be a way in which we are increasing that rate of mutation. Another one would be like a biological mutagen, so things like viral DNA. That viral DNA being inserted into the genome is going to affect that base sequence and therefore affect the rate at which the mutations occur. So I hope you've liked this video. If you have done, then please do click on the like button and subscribe to my channel. Also, if you haven't already done so, please check out my revision platform, www.aiqchat.com. It will help you in testing and tracking your progress.